to Janine Low-Marchelli. She received her degree in PhD in biomedical sciences from University of University of California, San Diego. During her postdoc training, she also received a K-12 award, which gave her advanced training in scientific teaching. Since 2020, she has been managing the technical information services for Jackson Laboratories. And as we're finding out is the importance of having good models to do the work, she's going to talk to us today about Jackson's efforts in the PDX area.
So this session is about big databases, and you'll notice that database is not in my title. I'll address that in just a few moments. My role as a technical information scientist and the team that I have is to really support your research and to make you aware of a lot of freely available resources, as well as any kind of ways that the Jackson Laboratory can support you in your research. So I connect really strongly, as mentioned in my bio, to the educational aspect of what our organization does. So our mission, if you are unfamiliar with the Jackson Laboratory, we are a nonprofit research organization, and our mission is to discover precise genomic solutions for disease and empower the global biomedical community in the shared quest to improve human health. So I think this mission can resonate with many of you here who are trying to develop therapies for oncology. And as I mentioned, we're a nonprofit organization. We're actually very unique. Many of you probably don't realize that we do have research faculty based in Farmington, Connecticut, as well as Bar Harbor. They are very interested, and they are focused on a number of different diseases, but oncology is one of the primary examples, especially in our Connecticut facility. My group represents the discovery aspect, so many of you probably know our mouse models that we provide to the research community at large, but I'll also be talking about the patient-derived xenograft resource that we also house and also provide worldwide to researchers such as yourself. And then finally, while I'm not officially part of the education department in our organization, I connect very strongly with that mission, and of course, that's why I'm here today. So as I mentioned, database is not in my title, but I did want to say that because this is leveraging preclinical databases for drug discovery, I did want to point your attention to a database that all of the data from the patient-derived xenograft resource is located in, and that is called the Mouse Models for Human Cancer database. And particularly, I will be showing you data from the patient-derived xenograft resource that JAX has established and has made publicly available to the research community. It's all very, very nicely described in this publication here, what this Mouse Models for Human Cancer database is. The focus is primarily GEM models, so genetically engineered mouse models, as well as our patient-derived xenograft resource. And I'll also point out that Array Express is where we have the raw data for all of our patient-derived xenografts. So all of you who know how to manipulate data, this is another source that you can use to get RNA sequencing data and microarray data from a number of these models that I'm going to present today. And I'd be remiss in not mentioning these other databases and these other resources that are related to patient-derived xenografts, which would be a larger consortium of patient-derived xenografts, which is the patient-derived cancers models, where this group is trying to collect all of the data and locations of different PDX that exist around the world, which also includes those that are available from JAX. Also, I'd like to point your attention to the National Cancer Institute resource, which is the PDMR. This is a really wonderful resource, very rich with protocols and best practices in establishing PDXs. So if you are connected to a number of these hospitals here and you are trying to create your own patient-derived xenografts, this is a wonderful resource that has very standardized protocols, very step-by-step protocols to engraft, maintain, propagate, cryopreserve, and so on. And then last, another database that I would love to point you to is the JAX Clinical Knowledge Base. A number of you in data sciences will be very interested in this as well, because it has a number of the actionable mutations that have been curated to this particular database, coming from literature, coming from clinical trials. So a lot of clinicians like to use this information to understand what's already been published about individual gene variants. And so coming to the Jackson Laboratory, something I did not realize that is a very core mission and a core piece of what our organization does is, it sounds kind of boring, but we standardize a lot of our nomenclature, and we standardize a lot of our information into easily searchable databases. And that standardization, I learned more and more, is a key foundation for reproducibility. So I'll mention that saying a number of people have brought up during the sessions today that the quality of the data that goes into your database is really important. And this is something that I'm very proud that our organization focuses very heavily on. So a number of you probably are in biomedical research for oncology, and you're eventually looking to put your therapies into mouse models so that they can then go on to clinical trials. And so I'll just quickly mention the difference between cell line xenografts and patient-derived xenografts. And I've outlined some of these comparisons here. But generally, what I want you to understand is cell line xenografts are very easy to manipulate. They've been around for decades. They are very easy to, they're predictable within a cell line. You know exactly how quickly they're going to grow. And overall, the costs are very low. You can manipulate them and add or knock out genes at will. So in contrast, patient-derived xenografts, they're understood to be more translationally relevant. They're more heterogeneous, and they're variable across different passages within the PDX, within the patient that it came from. And overall, they're a lot harder to work with and very resource intensive. And I brought my notebook up here, so I wanted to make sure that I got Dr. Al-Lazakani's name correct. She mentioned that something that we do in oncology is that we fail drugs very late in the drug development pipeline. And I'm just going to propose that PDX are another tool to help address that perhaps you can actually fail your drug sooner by using PDXs because they are so heterogeneous and because they are challenging and because they actually represent the variability that's seen in patient populations. So take that into consideration. PDXs, they themselves are not a perfect representation of humans because, first of all, they had to be associated with a hospital that had surgical tissue that was left over after what was needed to treat the patient to then make it into a mouse model. That mouse model had to be is its own barrier. So some of those mice, some of those patient tumors don't actually engraft into the mouse model. And that's dependent on a number of factors, including how the engraftment was done. And then the final kind of restriction is that tissue then has to be able to be propagated further in more mice. And that's shown in this collection side of how JAX established our resource collection starting in 2009. So those patient tumors were collected directly from the operating rooms, and I mentioned prior that these were tissues that were not available or not needed for the health of the patient, and these were considered surgical waste. And so that was collected and brought to our facilities in Sacramento, where they were then engrafted subcutaneously into the flank of an NSG mouse. During that process, there's a lot of quality control that is very well described in the references or the results and the methods section of this paper that was published late last year. So aside from kind of general safety, quality control measures, a number of quality control measures to ensure that human tissue was being grown out as opposed to mouse tissue, ensuring the identity of the patient sample did actually match the identity and the fingerprints of the tissues that were in the mouse, looking at histology, making sure that those cells were actually growing with the human Ki67 stain, and a number of other quality control measures that are described really well in this paper. So you'll notice that there are several rounds in which patient-derived xenografts don't actually make it into mice. That's represented by these gray bars, and then also by these blue bars. So these green bars are the ones that are actually able to become the experimental tools that many of you might be able to use in the future. You'll notice also that we've done a lot of genomics, and this is where that raw data lives in the Array Express that I mentioned. And keep in mind, this was starting to collect in 2009, and so there is a bit of a technology difference that you'll see some of our data has microarray data and some of our array data is from RNA sequencing. So it's with these that we can make these early passages of these tumors commercially available for drug testing. Long ago, the idea was these patient-derived xenografts would be the perfect personalized medicine. Wouldn't it be fantastic if a patient could put their tumor into a mouse, that mouse could then be used in studies and then could inform what types of drugs could be actionable on that patient? The reality is the timing doesn't work out, and a number of these models don't engraft. And so big data is really where these patient-derived xenografts will actually find their value. So our PDX tissue types that we've established, there are 364 of those models that we've established in NSG mice, and you can see a sampling of the different tissue types. Some of our early collaborations were with a very focused lung cancer team, so there are a lot of lung models, as well as colon, breast, bladder, and so on. And so the NSG mouse was selected because it is a very permissive mouse model that lacks B, T, NK cells, and hemolytic complement, as well as has reduced functions in dendritic and macrophage cells. So that makes it a very permissive environment for human tissues to engraft into them. And that very last point here, also CD34 or hematopoietic stem cells are also higher to engraft in this model because they contain a SERP alpha allele that's more similar to human, and that will be important in the later slides. So something that was important when we were establishing this resource was to choose the right mouse model. And back in 2009, when this was established, the best mouse model at that time was a BALB-SCID model. And what you can hopefully appreciate here is the BALB-SCID model was actually not very good at engrafting human tissues, and that the NSG model more faithfully recapitulated what the patient tumor looked like from a histological standpoint. And if you actually stain for those molecules, you can see that the primary tissue that was growing out is human as opposed to mouse. That's repeated here. You can also see that in these models, you can still get a reflection of epithelial markers and again of human Ki67 that you are propagating human tissues. So this is the database as it stands here. You can link to it from this link below, and my slides will become available later. And then you can navigate to this specific search. So some of the data that I mentioned here is target expression. So this is just a screenshot showing on the right amplification of RNA. Sometimes there's 2 bars, so you notice sometimes it has the patient data and sometimes it has the propagated tumor in the mouse model, showing that there is a nice correlation there. You'll also be able to search by gene variants. So the molecules and the variants that are displayed on this database were actually space constrained, so it's actually only showing a subset of all the data. As I mentioned before, all the raw data is deposited in a different database. And so what is searchable here, easily searchable, are common gene variants that are well annotated to the literature to be found in human cancers. You can also search for the model type. So this is just a filtered search for triple-negative breast cancer, but you can also search for the tumor type, the diagnosis. You can also look for characterization data. So we have about 3,000 images from histology. This is just one from a breast cancer showing that the passage 5 tumor is still stained for Ki67, and that this particular model is ER positive. There's a number of other kind of characterization data that are available. So some of these models were subjected to standard of care dosing once these models made it and were established into mice. And you can actually get that growth rate criteria as well as the response to standard of care, which I'm going to show here. So this is a really nice demonstration where there's a normal untreated mouse here with an untreated tumor that has a particular growth rate. But you can also toggle on and toggle off the other different treatments that were used in this particular model. And probably the scary thing is actually when you see all the data together, how messy PDXs are. And I actually really love this because it does highlight the importance of the tumor heterogeneity that's present and that should be expected and should be considered to be an advantage of using patient-derived xenografts. So that tumor heterogeneity may be more reflective of the human tumors and facilitates that study of variability of a response to a therapeutic before it actually gets into a patient. So I'll now describe how you can use the gene expression data. So this is just a proof of concept that says that you can look at the gene expression data, and based on the gene expression data, you can actually predict drug susceptibility of what's been published and what's known. So based on this particular tumor, this was I believe, a bladder model, and noted on its RNA sequencing was that there was no EGFR variant that was annotated and that it was low in this DNA repair gene ERCC1. So based on those characteristics, very simple characteristics of that gene expression analysis on this patient-derived xenograft, it would be predicted to be resistant to erlotinib, which is a EGFR inhibitor for constitutively active EGFR, and it would be sensitive to cisplatin. And so when we did that analysis in the lab, that was actually shown to be true, that under vehicle treatment, the erlotinib followed a very similar growth pattern as the vehicle treated, as expected by the lack of EGFR variant, and sensitive to cisplatin. So a more complicated example, a very interesting one, this diagram didn't make it into this publication, but it has been adapted from that publication, is an example of a particular patient who had a lung adenocarcinoma with an annotated EGFR L858R mutation, which would signify that this person had an acquired resistance to EGFR inhibitors. So that is basically what was happening here in the patient, shown along this kind of cartoon diagram, a timeline over the course of that patient's clinical history. So that patient had been diagnosed, had given chemotherapy, and started on erlotinib, in which they actually did see an initial response to erlotinib. Unfortunately, the tumor progressed later on, and this is actually when the patient-derived xenograft was, or the tissue was collected and the PDX was established at JAX. The patient continued to progress on erlotinib therapy, developed brain metastases, and eventually was given radiation and afatinib and cetuximab as a combination therapy, and that is where that patient then finally resolved. So we repeated that same drug dosing in the PDX that was established here, and it did mirror that response. So you'll notice here under vehicle conditions, there's the growth rate of the tumor untreated, and then in red is the erlotinib, which interestingly has a delay, but then does rebound with the tumor growth. And then, similar to the patient, was responsive to both cetuximab and afatinib. So what we can conclude from, or hopefully we can conclude from this, is that being able to establish the PDXs in these mice does give us a proof of concept that you can actually make predictions from that data, and that it can be validated in an in vivo system. So the last part I'll get to in the last 5 minutes that I have is how these models can be combined with humanized immune systems. So this is just a diagram showing that in one of my previous slides, one of the columns when I compared CDX to PDX is that eventually as time goes on, the patient's immune cells are not going to be carried on, and that's true for all CDXs and PDXs that you're going to engraft into a mouse system, is that those immune cells eventually disappear. So, or maybe don't even come with it. So this would be a way to address that gap if you're trying to understand the influence of the immune system. So we also have developed a platform in which we irradiate these mice and then allow them to engraft hematopoietic stem cells from cord blood. Those mice then develop a humanized immune system, which then can also be layered with a tumor, PDX tumor, or a patient-derived tumor, or a cell line. And so that is a humanized NSG mouse that has a human tumor. So the first question that many people had back when we were establishing these models and when others were establishing these models is that if you know anything about immunology, you wouldn't predict that these two cell types would be able to engraft in the same mouse because they are coming from two separate patients that have different HLA. And generally what we found, and others have also found, that if you've already got a patient-derived xenograft or a cell line that is engrafted into a mouse, it's probably a signal that it's a tumor that's pretty hardy. And also, when you're trying to, it's also probably a tumor that's been able to avoid detection by the immune system. So that when you actually put these two cell types from different patients into the same mouse model, a lot of the HLA mismatching is well tolerated. So this is just a diagram or some graphs showing that the overall growth rate of that tumor is relatively similar for the PDX on its own, as well as in combination with different humanized immune systems. That's not always true, and it's not always true from one donor to the next. It's also not always true from one mouse model to the next. So we do recommend that if anyone's trying to make these combinations of these very sophisticated models, that they take into account that variability, again, is a powerful thing to incorporate into your studies. But in order to not lose experiments, we also recommend using variable donors as well, so that those donors are equally represented in their response to different chemotherapeutics as well as immunomodulatory therapeutics. So how does this actually work? Does it work? It does. At least in most cases it does. So those immunomodulatory therapeutics do and can demonstrate efficacy in this type of platform. And shown here is one of the breast models here, just a standard of care cisplatin that you can see that's reduced, but also Keytruda, which is blocks PD-1 on the T cells and then prevents the interaction with the PD-L1 on these tumor cells. So the PD-L1 surface expression is, for this particular PDX, is described here. So this can show that you can remove that inhibitory signal on those T cells, but that the mechanism is probably allogeneic in this case because, as I mentioned before, the donors are two separate donors. So it does provide evidence that you can study certain immunomodulatory therapeutics in a model like this. So what I hope to summarize for you is that those of you who are really focused on data, there are RNA sequence data from these patient-derived xenografts from patient tumors that the Jackson Laboratory and others around the world have gone through that very difficult and resource-intensive expenditure to ensure that those can make it into a mouse model. So if you're very early in your drug development pipeline, you can use this data to select different models that eventually could be used for 2D or 3D culture that you know eventually will actually be able to be engrafted into a mouse so that you can get preclinical data before going into the clinic.